Welcome to another episode of Whose Gene Is It Anyway? I'm your host, Justin. As always, welcome back. I think this is gonna be a fun one. I've got kind of a fun thing planned for today. So, as you know, the way this show goes is it's basically centered around designing interesting DNA that does interesting things. Now, today we're not going to be pulling an idea out of a hat because there's something I feel like working on, and frankly, I think it'll be more fun than anything I can pull out of a hat today. So, um, it's okay. So here's the idea. First of all, where I got this idea is from one of my favorite books, a book called Walk Away, by Cory Doctorow. Which, if you haven't read it, fucking get on it because it's a great book. And so in the book, and this is not the center of the book, this is one of those things that it's just a really cool piece of science fiction that's sort of briefly mentioned, and that's it. Like it's glazed over very quickly, but it's a very cool idea, and so I wanted to see if something like this is even possible. So the basic concept is, at one point in the book, the main characters meet up with this group who feed them this stuff that is essentially food that's programmable by the light signals that you send it. So, like, if you give it a pattern of like red light, blue light, green light, red light, it might taste like one thing, whereas if you do a different pattern of lights or a different color of light or whatever, it would taste like something else. So obviously we're not going to be able to do the like intricate interwoven textures and tastes and all this kind of stuff. We're going to do a really stripped down version, but this is an excuse to talk about optogenetics and optogenetic transcription regulation, which is basically to say using light to trigger the synthesis of various proteins and interacting with DNA and constructs using light. Because there's all sorts of different ways to trigger DNA to turn on and off. Mostly they're chemical based, so you apply a specific chemical and it makes the gene turn on. And so one of the really common ones is what's called the Lac Repressor. So it works on lactose. So if lactose is present, it triggers the gene to turn on. And we'll talk about that a little bit. But what we're gonna do something really, really stripped down. Oh, thank you, Nico. Thanks for kicking us off. Speaking of which, you'll notice the donation bar, doobly-doo, thingamadoo. Nothing specific today. It's just for general lab supplies, tubes and tips and all that kind of stuff. Just the usual things that keep the lab running. Or, food, because I've been told that's a thing you're supposed to eat, like, twice? 3 times a day? I hear that's important. Anyway, so I also have some other updates, which we'll talk about in just a moment. But yeah, that's the basic plan for the day, and I think it's going to be kind of fun. So you may notice the stream starting a little later today. I was actually just out getting some more plants. As longtime viewers will know, I love plants. Plants are wonderful. As you can tell behind me, like here, this is one of my plant grow areas, and so I'm always looking to round it out and add more, but also occasionally I get things like fungus gnats and other insect pests, and so I was out getting marigolds, which is a type of flower which is very well known for repelling insects. So I'm basically going to be spreading them around my various grow areas to try and repel them. And I also got a few other things that I like plants, man. Plants are wonderful. So I got some tarragon, I got some lantanas, I got a passion flower, bunch of interesting things, and so yeah, that's gonna be a lot of fun. So basically after the stream I'm gonna be doing some more planting, which is gonna be real fun. I just got some more strawberries. My plant area is getting wonderful. I'm probably gonna, so I'm actually planning, just for those who are wondering, because I do talk about plants a lot, I will actually be doing a video about my various plant growing, they're not even experiments, just things that I do. I'm inherently lazy, so I'm going to be using robots to water my plants because I fucking can, and it sounds fun, and it's something to do. So yeah, I'm going to be robotifying all of the plants on all of the different shelves and all of the different areas. So I'm going to be, the first of the little Arduino-based kits is going to be arriving this week, and so I'll be starting to film that soon. It's going to be a lot of fun. Also, for those who have been asking, there will be a new Taste Emporium video very soon, this week. I'm literally just waiting on a piece of equipment to arrive, which it's supposed to be here today. And then basically for the next 2 days I'll be filming, and then I'll be editing on Thursday, and the video should hopefully be out Friday or Saturday. So that should be a lot of fun. If you have no idea what I'm talking about, the Taste Emporium is my second channel, and you should absolutely go subscribe. Speaking of edible food tasty things, in actually the very first episode of this series, we made 2 plasmids. And actually, you know what? I'm going to switch to, since we're starting to get into it, I'm going to switch into my show desktop view so we can see that. So in the very first episode of this series, we made these 2 plasmids. If I can find them, here they are. So one of them, so these are the 2 of them. So I'm going to open them up really quick just so I can show you what's what. So this is the first one. It says how can MX. Oh, thank you. Well, thank you, Mr. Roberts. Greatly appreciated. Or MD Roberts. Is it Dr. Roberts? I think I've called you Mr. Roberts every single time, but is it MD Roberts? I'm gonna just call you Dr. Roberts because that sounds fun. Anyway, so this is the first plasmid that we made. So it is a plasmid that works in Saccharomyces cerevisiae, which is also known as baker's yeast, and you'll see that it's got this little chunk here, which I'll zoom into so you can see it a little better. Oh, who just? Well, thank you, Shoot Raw. Greatly appreciate it. So I'm going to just zoom in so you can see this a little better. Oh, I gotta scroll the other way. So you can see this here. It says ovalbumin. For those of you not familiar, ovalbumin is egg white. So this plasmid would make egg whites in Saccharomyces cerevisiae. And we may actually be using something like this in today's construct, which is one of the reasons I wanted to highlight this. But importantly, I have said this in I think the last several streams that I've been waiting on a quote, because we did a fundraiser, and the plan was to actually get this and the other one I'm about to show you actually made. And so I finally heard back from the company. And, okay, you're getting fucking, that's getting really low. So I actually heard back from the company, and they're actually going to be making this plasmid. They sent me the quote, and it was exactly what I expected. So for this plasmid, it came out to, I believe, 430. And then for the other one I'll show you, it came out to 1,200. But then there was a 20% discount, but then I also have to add an extra 300 for some of the extra plasmid prep that I want to make sure that I never run out of the DNA. Somebody just kicked in. Oh, your name is very, very long. Well, thank you, anonymous, extremely long string of numbers. Greatly appreciate it. But anyway, so I finally got the quote back, and so basically the only thing that I'm waiting on to basically tell them to like get going and start making this is this plasmid, which is the actual empty backbone that I ordered from Adgene, and we'll talk about Adgene at some point today probably. But think of it as sort of GitHub for bio, but you pay. So, you know, they carry all these different plasmids, and you can buy them from them. So this is the empty plasmid. We ended up making this one. So this one makes egg white. This one makes all of the proteins. So this one you'll see is obviously much larger. So if I scroll, you'll see deer beta-casein, kappa-casein, alpha-lactalbumin, beta-lactalglobulin. So these ones are deer milk. So it's milk, but I took these proteins out of white-tailed deer because they're one of the only species that hasn't been patented. So this plasmid, as well as the egg plasmid, are officially in the process of being made. Once my empty plasmid arrives, I can send it off to the company so that they can actually synthesize the DNA, stick it into the plasmid, and then send me the finished plasmid. And then we can put it into some yeast and make some weird egg and milk and grow myself a cheese omelet. Omelette de fromage. Anyways, so yeah, that is the plan. And I'm super excited about that. Thank you everybody who chipped in for those, and all the people who chipped in. I will make sure that you get a stab of the finished plasmid so you can play with it. And eventually, once I get the yeast actually modified to contain this, then I'll be sure to send that out as well. Still haven't decided if I'm going to host this in a store, but I'm definitely going to share this on AdGene, so anyone who has an AdGene account will eventually be able to buy this and play around with it. That's going to be a lot of fun. So, that's my update. I'm very excited about that. I've been waiting. So it turns out the reason that it was taking forever to get a quote back was they had changed their email settings. And so the email had gotten put into spam, and so we had to call the company and be like, hey, did you get our email? And they're like, what email? And, they found it. So now that the emails are getting through, I got a quote within a day. So that's really fantastic. Super excited about that. Egg and milk getting made. Super excited. We'll keep you updated on that. Okay. So I'm going to just take a moment and have a look at the chat. Sorry, I got the hiccups. I'm going to have a look at the chat, see if I missed anything, and then we're going to get into the optogenetic goopy food stuff. And admittedly, the thing we're going to end up making today, I mean, there's a chance that we end up designing it to work in E. coli just for the sake of... Excuse me, I really have really bad hiccups right now. So yeah, there's a chance that we're going to design it to just work in E. coli, so it's not really going to be food safe, it's more of just like proof of concept. But we may end up doing it in yeast, so we'll see. I have some papers already already pulled up to basically have a list of optogenetic systems that we can work from, and so we'll have a look around and we'll see what we can find. Okay, let's have a little look at the old comment section. Alright, let's see if I missed anything interesting. Okay. See if there's anything interesting. Somebody asks, how can you even patent the DNA for milk of animals? So it sort of depends on the country. Some countries don't let you patent nature, so you're not allowed to actually patent natural sequences, but you are allowed to patent the use of them. So while you might not be allowed to own the sequence for cow milk, for example, you could be allowed to patent the use of transgenic organisms, so like yeast, to produce the cow milk proteins and then use them in a very specific situation, like say making a synthetic milk. So it's really, that's more of a, it's the sort of thing that with a talented enough patent lawyer, you can either break or fight most patents when it comes to bio stuff, just because certain things are not allowed. But then you end up having to deal with court. It's just not fun. Do you speak French? The cliche omelette du fromage was weirdly well pronounced. Well, I mean, I live in Quebec, so I should speak French. I don't, but yeah. It's one of those, I've lived here for a while, and I really should get around to learning French. And it kind of I'm slowly picking it up by osmosis, but everyone I know speaks English, so I've had no actual need to speak French. Other than when, a grocery store attendant, hops into, French at full speed, and I just kind of, smile and nod, and, yeah, until it's clear that they're expecting an answer, and then I go, English, please, help. But yeah, so that's that. Somebody says, what did you study to know how to do this? I studied biology, technically, but I dropped out of boredom, mostly. I've talked about this a million times. I'm not going to get into it right now. If you want to learn how to do this, you just start doing this. There's very few actual universities that will teach you this stuff. I mean, you can try taking a biochem major if you want to try and learn this stuff, but I mean, depending on the exact program, that could take you into a million different directions. If you can find a synthetic biology course, that's the way to go, but I mean, those are few and far between. So really, the best way to learn how to do genetic engineering like this is to just do it. And fail, admittedly, a lot, but that's how it goes. Okay, one more question and then let's get into this. Do you know a different alternative to Genome Compiler? I am literally using that thing right now. It's called Benchling. For those of you who are just kind of catching this for the first time, this website I'm using, it is called benchling.com. It's normally known as a note-taking site. Well, not like, you know, like homework. We're talking like notes for like lab notes essentially, and it's what I've used for taking lab notes for a very long time. But they recently essentially made their own version of Genome Compiler, in like and built it into their site, and so it's what I've been using. And honestly, I've come to really like it. It's definitely taken some getting used to, and there's some like definite quirks to it, but it works really well. We're not doing any CRISPR stuff today. We'll probably do CRISPR stuff another day. I'm not feeling it. I really want to do optogenetic stuff, and there should be absolutely no reason that we need CRISPR for that. Here's a really annoying question that I'm going to answer anyways, just because yeah. Why don't drug cartels use this technique to mass produce their product cheaply? Because doing this is hard, and it's also a lot easier if you have a whole frickin' forest to just grow the plant. This, like, case in point, I have a friend who has a company that has made a strain of yeast that produces THC. It has cost them 10 million and taken them like years and years and years to get to the point where they're even making enough that it's even worth growing. So it's very difficult to actually like make something that a cartel would be interested in via genetic engineering. Like, it's possible, sure. It's very hard, and there's easier ways of doing it. Primarily, grow the fucking plant. There's already infrastructure for that. So yeah. That. Okay, cool. So let's actually get into this. So the first system that I want to talk about is this one. It's the P-Dusk and P-Dawn system, which comes from this fantastic paper called From Dusk Till Dawn, 1 Plasmid Systems for Light-Regulated Gene Expression, for those of you who'd like to follow along. Sorry, my phone's going. I just want to see if this is anything important. I assume no. No. Okay, cool. Wait, what was that second thing? Yeah, okay. I can deal with that later. Cool. So, anyways, this system is really cool. I actually have this one. I have this sitting in my fridge, and so I'm actually going to be doing a video on this system, like a proper video, not a stream, on this system soonish. So what this system is doing, and we'll talk about the exact way that it does this, is it's essentially a light-triggered gene expression system. So P-Dawn, for specifically, when it's exposed to blue light, I believe it's 470 nanometers-ish, like in that sort of range, from like 460 to 480-ish. So that's like, actually it's about this color of blue, which, you know, they actually did a really good job writing this paper. When it's exposed to this color of light, it triggers it basically triggers gene expression. And we're going to talk about how it does that in a second. Because technically it's triggering when the light goes away. Because this is an inverting system here. So the difference between P-Dusk and P-Dawn, P-Dusk turns on when the light is removed, and P-Dawn turns on when the light is applied. So it's basically the same starting system, but they've got like think of like an inverting, like a NOT gate in electronics, essentially, is the closest thing that's equivalent-ish. So basically, we'll talk about how it does that specifically in a second. So I'm going to scroll down a little bit, and I want to see if I can find you a picture, because I know the picture is in the supplementary data, which I'm going to have to probably pull up separately. Ugh, gross. I'll save here. Yeah, here we go. So, I mean, this is oh wow, that's where he got that image. Exciting. Pfft. I like when you click download full-size image and it's still tiny. Okay, so this is going to be a little blurry, but you get the idea. So with P-Dawn, this is no light. Oh, there's a fungus gnat. I think I just swallowed a bug. Give me a second. I need to get water. Well, that was really gross. gnarly. Um, oh, yeah. Airborne protein, not exactly fun. Oh, somebody kicked in. Random forest. Should I quit my minimum wage job or save up for a PC for deep learning? I am still studying. I mean, I don't just quit your job unless you have another income source. Sounds dumb. I don't know. Anyway, so yeah, this is p-dawn and p-dusk. So you can see p-dusk turns off when you expose it to light, and p-dawn turns on when you expose it to light. Also, what you can see is p-dawn ends, and I mean, it's hard to know if they took these pictures at the same time, but my understanding is, and even from what I remember from the paper, p-dawn works better. So you get way higher levels of gene expression. I don't remember why, but that's just the way it is. So yeah, p-dawn, p-dusk, very exciting, very cool paper, very cool system. So how exactly does this work? Now, again, I'm going off memory because I don't want to have to like read through this whole paper again while I'm live. But my understanding is these two bits here are the actual light-sensitive parts. Okay, okay, so... Well, somebody kicked in. Thank you, Delene. Bug compensation. We found a bug. Anyways, so... yeah, okay, so YF1, I believe, is the actual light-sensitive part. So when it is triggered by light, it drives gene expression of this fixed J, which I believe triggers the actual gene expression of the thing that you want. So there's a promoter in, like, over here, where you would put your gene of interest. So you'd put your gene of interest here. So MCS is multiple cloning site. So you put your gene of interest that you want it to express here, and then when light hits the fixed J triggers this thing and makes your gene. For p-dawn, there's a little bit of an inversion here. So the light drives a repressor. I believe that's what this is. So, or maybe it's the other way around. When there's no light, this doesn't get made, and there's a repressor here. So when there is light, it turns the repressor off, and so you get gene expression. So it's a bit convoluted, but it is a very cool system, and it works pretty well. So yeah, that's p-dawn and p-dusk. So I think we could use. I mean, I think we definitely could use this. The only thing is this only works in E. coli, as far as I know. So I don't know if it would work in something that's more food safe, like a lactobacillus. So that's a bit annoying. It could, though. I honestly don't know. I don't know. I don't really have good data on that. But there's also but this is just one example system of a thing that can do this. So this is a more complete list of the various systems that can be used for this, and you'll see the light and the activation wavelength. So most of these either work on red, blue, or UV. So there aren't really a huge number of systems. There's like not a huge amount of variation here. Also, when it says like 450, 474, like it's kind of that thing. Like it's not necessarily exactly that wavelength. That's usually just the peak. So there's usually a fair bit of bleed on either end. That will actually trigger the thing. So yeah, that's kind of our options. So what I'm thinking is if we had a red and a blue, then we could drive two different things separately, essentially. So what we really want is something that would work in like food safe stuff. So I see one. So this one is a yeast. So it's 5e PIF. It's PIF3. Don't know what this is, but we'll look at it. So this one would drive expression in yeast, so that would be good. Now if we had a red one, so here's another yeast one. This one's called Tulip. It's so cute. So this one would drive it at blue, and then I don't. So yeah, this is weird. So we've got a red and a blue. They would both work in yeast. So we could build this to work in yeast pretty easily. So let's see if we can actually find some of these. Because if we can't, then we're sort of shit out of luck. First things we're going to do is we're going to actually look on AdGene, which I mentioned earlier. And we're going to see what they've got for optogenetics. Because really, that will make my life a lot easier. Any interesting ideas for a biochem project for a hackerspace in the EU that can't legally do genetic engineering? So you can legally do genetic engineering, you just can't do the type of genetic engineering that freaks the EU regulators out. You can do mutation stuff for plants. So like if you expose them to like UV, you can get plants to mutate. You can do plant tissue culture entirely legally. There's no laws against it. You just can't do any like genetic modification, but you can do all kinds of very interesting biochem. So if you learn how to do like plant tissue culture, for example, you can do like protoplast isolation, protoplast fusion, flower induction, making like interesting crosses that way. There's all kinds of stuff you can do. But if you want to work on GFP, they kind of shit the bed and freak out. So that's sort of how we go. Okie dokie. Let's see what we got here. Lentiviral, mammalian, lentiviral, AAV, mammalian, lentiviral. Yeah, this is all meat. This is not really what we want. Let's see. So this is a different. So there's two kinds of optogenetic. There's optogenetic transcription or gene expression, which is what we're interested in, and then there's optogenetic like nerve triggering, which is something that there's a huge field of research now where they'll basically like genetically modify the brains of various organisms, usually rats, and then they'll basically implant like a fiber optic coupler so that they can shine light on very specific parts of the brain and essentially just like trigger it on command by just like shining a little bit of light on it, which is kind of neat. And so people have done all kinds of very interesting stuff with that. So this is all mammalian and worm, and so this is for working in flies and cells and that kind of stuff. Not really what we want. This is more what we're looking for. These are the optical switch plasmids, and by the looks of it, there is a lot of them. So let's see what we got. So I saw a tulip, optical switch tulips. So let's see what this one is. Because so Phi Piff was also one of the ones that I remember from that list that we were just looking at, so that's very cool. Okay, so I haven't actually looked at the comments a little bit, see what we got. So the incubation time, this is, it should trigger in about an hour. So if, actually, it was in this paper, I can show you. So this is, time at expression conditions. So if this is 5 hours, it's like at peak expression within half an hour to an hour, basically. No more than 2 or 3. So it really doesn't take long. And so, you give it its exposure and then you just let it grow normally. And that's enough to have triggered it. So in this case, like you would want to keep your culture in the dark, essentially. And then trigger it for, half hour or whatever, and then let it do its thing. Okay, so let's see what these plasmids are. Oh, S. cerevisiae, that's fun. That is exactly what I like to see. Yeast expression, urease 3. This is the tulip one, right? So tulip is blue. Multiple cloning site, TF promoter. Yeah, but where's the optical stuff? Tulips. Tunable, light-controlled, interacting protein tags for cell biology. Let's have a look at this paper, see what's up. Tunable, light control. Man, somebody definitely like took some time to figure out the right words to call this tulips. Let's just be really clear about that. Biologists are nerds. And so they totally do shit like that, and I love it. It's a DOI of N-meth. Yeah, that sounds Delicious. Is the, where? Ooh, I don't even need to Sci-Hub this. Free articles. My fave. Let's see what we got. Photo switch design. So by the looks of this, it's like a weird... Oh, wow, this is weird. Oh, this is so weird. Okay. So it's like a conformational change to the protein that makes it do the thing. That's neat. Okay, yeah, but what is this? What is this plasmid? Where does it... PDS 221. Let's see if that's actually referenced in the paper. Yes. Oh no. 221? No. Okay. Well, balls. Okay. Well, let's see what else we got. Oh yeah. See, anytime I see something like this, I'm just like, oh shit, because you know it's gonna be just a nightmare. Let's see what we got. This is interesting. I mean, it's a little hard to parse live. Making engineered photoreceptors, we reason that a fusion of peptide epitope to the C-terminal of the JA helix, oh my god, allow the love interaction to sterically block or cage the epitope from binding to... Okay. What does that even mean? Okay, peptide thing, love to... I'm confused. Yeah, this is super weird. The question is, what else is in it? Is there another map? Oh, there we go. Okay. Yeah, there we go. So it's this EPDZB1. I assume this is the interesting light-sensitive bit. And I assume there's a multiple cloning site right here, which would make sense. It's interesting that they're just using the TEF promoter in C1 Terminator. So that's very weird. Not entirely sure how this would work. I'm gonna have a look at the other plasmids, and we're gonna see what else is in here. Wait, is this the same one? Are we looking at this twice? Yeah, we've got this open twice. Okay, let's see what this one is. Oh, it's poorly labeled. Also, this is CMV. So a couple of things. I know I'm just sort of rambling and circling at the moment. I'm trying to parse a lot of information. I knew this one was going to be really hard, but I'm going to try and break it down as I understand it. Just kind of bear with me if you can. So a couple of things. As soon as you see CMV or CMV enhancer, promoter, whatever, instantly you know that this is a plasmid for working in animal cells, which for our purposes is useless. Because while it would be interesting to make, you know, beef that like tastes like, you know, grape or whatever, when you expose it to light, not really what we're going for. Okay, plasmid type, mammalian, multiple, yeast. Here we go. Ooh, this one's a cry one. That's exciting. So cry2 I recognize as a photoreceptive protein from eyeballs. Is there another map that tells me what in the hell is in here? No? Okay, well there's cry. That's cool. How is this supposed to work, though? Oh, okay. What is this? This is like every plasmid from their paper. Yeah, this isn't what we want. Actually, you know what? I'm going to just open this up. If anybody can find a nice optogenetic plasmid for working in yeast on AdGene, leave it in the comments, and it'll speed these things along. Otherwise, I'm going to end up doing this for like 20 minutes, and that's not really fun for anybody. Okay, here's another one. Let's see what this is. Gal promoter, GFP, cap binding site. Yeah, I'm not seeing the thing that I'd like to see. Oh, there's the love fusion thing. I still don't know how this system works, though. It's a little weird. Yeah, because it's similar to this one. So where was that paper? Interesting. Okay, so I don't know if this really drives gene expression or when it. Okay, so looking at this, actually, now that I'm seeing this, I actually have two plasmids open, which, so you'll see this one says EPDZ, M cherry, whereas this other one is GFP love. So now if we look at the paper, I'm seeing GFP love and EPDZ M cherry. Cool, great. And by the looks of this graphic, when you expose them to light, they stick together. Now my question is, how does that help? Like, it's like, okay, cool, you can make the now they kiss whenever you expose them to light, but how does that actually drive gene expression? And my suspicion is it doesn't. So not really sure what to do about that, or if it's helpful, which I'm feeling like no, it isn't. It looks like some sort of protein cage, so it's like a way to, on light, you can make things stick to the like cytoplasmic membrane, but I really have no idea why that would be helpful. Okay. So yeah, I'm kind of leaning away from this tulip stuff because it doesn't seem super helpful at all, really. I mean, here's P dawn, P dusk. So we might just start with P dawn, P dusk just because it's easy, and I have it, and I could actually work with it. This is not helpful. Mammalian, AAV, bacterial, yeast. Here we go. This is another 5B one. Let's see if this is any more interesting. It is poorly labeled. I mean, it's chunky, so presumably some stuff is going on. But, I mean, I don't see a multiple cloning site. I don't see any way that you could actually stick DNA in here, so this looks like useless. We're going to keep looking. We are going to keep looking. Mammalian, yeast. More tulips. Okay, this tulip thing is really not helpful. Tulip. Not helpful. Let's have a look at this one. Oh, I did not need to do that. We're just gonna kind of look around, see what we got. This looks more promising, but again, I don't see a multiple cloning site, which is gonna make this very hard to work with. But there is another map. A admittedly poorly labeled one, but a map nonetheless. PGAL 4AD CIBN. What does that one do? Let's have a look. Let's look it up. I bet you AdGene has it. So what was it called? It was PGAL 4AD CIBN. There's only one. Okay. Seems very popular. But what does it do? Rapid blue light mediated induction of protein interactions. Yeah, this is the same problem that we were having before, where this is protein interactions. It's not actually protein synthesis. Okay, let's look this up. Yeast optogenetic gene expression system. Okay, here we go. A yeast optogenetic toolkit for gene expression control. There we go. That's all we wanted. Okay. Okay. What's this do? We reconstitute activity of a split synthetic zinc finger transcription. Oh, wow. They're using zinc fingers for this? That's fucking wild. Okay. So they've stuck a cry protein to... So this is using cry. Okay. So they've Okay. So this actually sticks to a specific promoter. It's 2 pieces. What am I looking at here? I need to make this bigger so I can actually read this. Okay. Assembly connector, promoter, coding sequence, terminator, assembly connector, marker, homology, marker, homology. Okay, so this is describing the basic structure of their plasmid, which is fine. So it looks like as long as you've got whatever this weird promoter is, it should drive the expression. So I think DBD cry 2, based on this little picture here, is, I assume, this chunk right here. So this bit will actually bind to a specific promoter, which is good. We want that. I mean, I remember them saying that this was a zinc finger. So zinc fingers are sort of like CRISPR's older cousin, essentially. So in the same way that CRISPR can target specific sequences in the genome, zinc fingers can do the exact same. It's slightly different. They work differently. They're harder to design, but once you've designed them, they work really well. So by the looks of this is a zinc finger stuck to a cry protein, cry being the photosensitive thing. And by the looks of it, this kind of sticks to the gene of interest, or I suppose the promoter of interest. And when you expose it to light, there's this interaction between this other protein complex and the cry protein. And so when they actually come together, only when exposed to light, it actually drives gene expression, which is what we want. So by the looks of it, you would have to express these 3 proteins all on one plasmid, and then have this one promoter here to basically be the spot where you could stick your gene of interest right here. And it should drive it when you get light exposure. And based on this chart here, it's not super speedy by any stretch, but after more than a few hours, you end up with this huge spike of gene expression when exposed to light, which sounds like exactly the thing that we want. So now the big question is, can we get this plasmid? Because that would be fantastic. Optogenetic strain is tunable as a function of blue light intensity. Yeah, okay, cool. So, wait, is that the end of the paper? Oh, for fuck's sake. Okay. Okay, so it looks like this is the name of the thing that we want. VP16 CIB1, which I believe was that second cluster. Yeah, here we go. VP16 CIB1 is part A, Cry2 is part B. So, big question. What is the name of this plasmid? See if I just look this up, if anything comes up. Okay, let's check Adgene. Yeah, so if anyone can find this thing on Adgene, do let me know. It would save me a whole bunch of time. I'm gonna look up ZDB. What was it called? It was, where did it go? ZDBD. ZDBD. Cry2. That's probably going to come up with nothing. That's also going to come up with nothing. Yeah, okay, let's just look up Cry2. See what comes up. Okay, lots of options. Okay, so let's go species. Saccharomyces cerevisiae. That'll narrow this down. ZDBD. Zubidibidi. What is this? This looks like it could be right. It's SV40. That doesn't look right. Yeah, I'm only gonna spend another like 10 minutes looking for this, and if I don't see the thing that I'm looking for, then we're gonna just switch to doing this in E. coli, because I don't want to spend the whole time looking for plasmids. It's just not fun. Okay, P rev this. I don't know what that is. Not what we're looking for. Not what we're looking for. Not what we're looking for. Wait, they gave this a name, didn't they? YOTK? See if Edgy knows that. Oh, hello. There we go. Integration vector for optogenetic machinery. VP16, CIB1, or yeah, CIB1, and Z-Cry this thing. That sounds exactly like what we need. Okay. There's your CIBN promoter. So, I mean, this looks right. Okay, so I'm gonna just open this in a new tab, and we're gonna just save that for a second. What was the name of this author? Anne Adricun. I'm butchering that name. I apologize, but I want to see if that's the same. Yeah, this is it. This is the one from that paper, or at the very least, it's close enough. In fact, let's have a look at this. Yeah, it is from this paper. That is awesome. Okay. Now, if I click this, there's a chance that it'll show other plasmids associated with this. Yeah, here we go. So one of these is going to be the thing from the paper that we actually need. Oh, for fuck's sake. My mouse is a little bit glitchy. So let's just have a look. Wait, oh, Jesus. What the fuck? M Ruby Reporter. P weak M Ruby. Okay. Wait. They provide a better map? No. Okay. I don't know. I'm not seeing a nice multiple cloning site, so that would make it very difficult to work with. I mean, these all say that they've got reporters, but I don't see them. It's a very cool system, very poorly labeled, which in my mind is the ultimate sin of any biologist. Label your shit. Oh, that is the wrong button. That is entirely the wrong button. I'm going to just keep opening these until one of them is either the thing that I want or I get bored. We're going to see which one happens first. Not labeled. Kind of labeled. No, not labeled. I mean, these all look identical. Like, there's no obvious difference between them. It almost looks like you gotta have two plasmids, which sounds very annoying. And then these are all the ones I've already looked at. Frustrating. Oh. 1, 2. Oh. See if that comes up. What the? So Nico asks, so are there naming conventions for DNA sequence? Are there naming conventions for DNA sequences, or do people just pick something like they like? Yeah, there's no real. I mean, like theoretically, there's a naming convention, but like, shrug. Oh, hey, Sassy, welcome as always. This looks interesting. Okay, there's CIBN. This looks like it's missing machinery, though. VP6. So this looks like half of it. Like, this looks like half the system. And then some of these other plasmids that we... Actually, you know what? Sassy's here. Maybe they'll be able to find it. Okay, so just as a review for anyone who's just tuning in, we're working on optogenetic systems today. So at the moment, I'm just trying to find a good optogenetic expression system, ideally for yeast, just because yeast are food safe. But it could also be in E. coli if that's all we've got. And it's basically sort of what we're going to be shifting to in a minute, just because I can't make heads or tails of this at the moment. So I think what actually might be really good is to start with just the P-Dawn and P-Dusk system, because I know that one, I know it works, I know how to find it on AdGene, and I know where you cut to actually make it like do the thing that you want. So if we start with that one, we can at least get like a prototype designed out where, you know, when you expose it to light, it does one thing. When it's in the dark, it does another thing. And so we, rather than having, you know, multiple different colors doing different things, we can just have a simple on-off. So, you know, we could do it if you grow it in the dark, it tastes like grape, or if you grow it in light, it tastes like Fruit Loops. Like, you know, we could do something really simple like that. And I think that could be a nice starting point. But since Sassy is here and they're a freaking god at finding useful plasmids, if they happen to find, I mean, and if anyone in the comments happens to find a nice expression system on AdGene or anywhere else, it doesn't really matter. As long as I can download the plasmid map and have something to work from, then we'll use that. But for, I think until then, oh yeah. So yeah, until then, I think we're just gonna switch to doing P-Dawn P-Dusk, because I'm frankly, this is very cool, but I don't think I can parse this information. So, just to be really clear here, this is one of the hardest topics that I've chosen, and I chose it for exactly that reason. I wanted a bit of a challenge because I was getting a little bit bored, and I thought, you know what, screw it, let's do something really hard. But I'm gonna review this, and we'll probably come back to this sort of system, this VP16 thing. We'll probably come back to it in a future episode once I've had time to like really sit and chew on the papers and like actually like internalize the information. You know what I mean? Okay, let's actually get into doing some stuff, because fuck this noise. So there's an empty one here, which should be this one. Yeah, there's the lambda repressor, and it's poorly labeled. Okay, oh fuck. We're gonna grab the GFP one because, well, I mean, then I at least know where the thing is, and what not to break. So actually, yeah, okay, let's download this. And I'm going to grab the empty version as well, and I'm going to close all this crap because it's not helpful. It's not the thing that we need. Yeah, the tulip thing is definitely not what we want, and the cry system is very interesting, but also I don't think it's what we want. So I'm going to close this as well. I'm going to save that for, because we're doing P-Dusk. Okay. Let's go to taste change goo. Because, you know, I'm an adult. I can name things how I want. Yeah, no pressure, Sassy. You've been very helpful thus far. Sean's Homes Up says, if we grow it in the dark, can it taste like fruit by the foot? And if we grow it in the dark, or if we grow it in the light, can it taste like fruit by the meter? No. Sophia, I'll give you the same answer that I've given on essentially every single stream. The resource that I always recommend to get started in this sort of thing is the Manga Guide to Molecular Biology and the Manga Guide to Biochemistry. And if you're unfamiliar with organic chemistry, I'd also recommend the Manga Guide to Organic Chemistry. read all of those, cram the information into your face hole, and then pick a project and start working on it. It's really the best advice I can give you, because anything else is just it gets so theoretical, and the only real way to learn any of this stuff is to just do it. Like there's no shortcut, there's no textbook, there's just pick a thing, do the thing. Because like it's one of those, once you've been staring down a project for long enough, like you start picking, like even working through one very simple genetic engineering project, like by the time you're done it, like the amount that you have to learn to actually make it work can be just massive. What is it about to call this? I'm going to have it call it this. Not that this is helpful. I'm going to rename it anyway. So we're going to rename this to, it's Codon, I believe. Okay. And this is the empty one. So this should be, oh, yeah, that's what I like to see. This is, okay. Terminator. Lambda repressor. Site. So this looks like the multiple cloning site, based on the fact that there's a shitload of restriction sites right here. But this is an important one. It's a little weird. So I'm gonna grab the superfolder GFP one. This is one I actually own. Like this is in my fridge. So I'm gonna grab this one. The only reason I'm going for this one is because this one has a protein that is already being expressed, so it's much easier for me to look at this and go, okay, this is exactly where you need to put a protein to have it work. Waldo, are you familiar with the S mutants gene editing that fights cavities? Yes, we have tried so many times to get that stupid construct to work, and at this point we're fairly certain that the researchers who did it were maybe full of shit, or I don't know, but we have not been able to replicate their results. Like the gene synthesis company that I use wasn't able to transform the bacteria. Like there is some shit wrong, and we don't know what it was, and it just doesn't work. Is manga really the best guide a knowledgeable individual like you recommends? That's kind of weird. Well, maybe get off your high horse, because it's actually an incredible way of, like, it's a great book. It contains a huge amount of knowledge, and it's presented in a very fun way that makes it accessible to the average person. So yeah, it is. Rather than like some dusty ass textbook where it's just throwing terminology at you and it's not fun. Okay, so did this download? Yes, it did. Cool. I wish it did drag and drop, but it doesn't, and it's so annoying. Okay, so let's import the green one. Should be this new one. Download. Let's close this. Rename this to Codon GFP. Technically it's superfolder GFP, but that doesn't really matter. Okay, I'm going to close this for now, just because I want to have a look at this and see if there's a nice convenient restriction site, which is what I'd like to see. So this should be our superfolder GFP. This is the thing I care about. Oh, that is really frustrating. Ooh, there's an NDI site. That'd work. Let's see how many of those are in here. 3. Shit. Okay, well, that's unfortunate. Where's the other one? I see this one. See this one. Where's this one? Oof. Woof. Okay, that's a problem. Okay, so is there anything at the end of this that would allow us to cut it out? Okay, XY. So I'm just kind of working quickly just so I can get a brief overview. Ooh, That's exciting. Let's grab that one. That's helpful. Let's... I just want to... Oh, there's only one. That's what I like to see. Okay, I'm going to turn all these other ones off because they're not helpful. Okay, so let me... Now I can actually... Now I've kind of got a handle on what's going on. I can actually give you a little bit of an overview of what I just did. Okay, so we imported our plasmid, which is our piece of code, essentially, into our program, which, as a reminder, is benchling, which is wonderful. So this can look a little bit confusing. I'm gonna zoom out so you can kind of see the whole thing in one shot. So unfortunately, some of this isn't labeled. So you'll see this like empty chasm of like what is here. So there's definitely like the other parts of the genetic circuit exist in this area. This is actually where all the interesting, useful, like optogenetic stuff exists. It's just not labeled for some fucking reason. And frankly, I just can't be bothered to try and label it myself. That's a task for another day, but it honestly doesn't really matter. The important part is this part. That is the lambda repressor. That's going to need to be... I'm going to need to change the color on that, because that's going to get really annoying really fast. Did it not save? It did not save. Of course it didn't, because fuck me. Okay. Save. Fuck's sake. Cool. Okay, so this green bit is the thing that we actually care about. All this stuff, like, from basically, like... Oh, no. No, come on, you fucker. There we go. So, basically, all of this is all the optogenetic stuff. So it's all the stuff that we talked about at the very beginning from this paper. It's not this paper, this paper. It's all these pieces. So it's the YF1, fix J, and the CL, because we're working on Codon. But you'll see the MCS that was here, MCS, multiple cloning site, and the PR. This is the promoter that is actually sort of the trigger. So that all exists over here. So by the looks of it, there's actually a fair amount of... Wait a second, why is there a his tag here? Man, I ran into this problem the other day where things started transcribing in places where I was not expecting them to, and it was super annoying. The thing is, there's a ribosome binding site here. T7 tag, thrombin site, his tag, fucking... Because the thing is, the gene could start here. Let me just check something. Because I had this issue last time, and it was not fun. Okay, no. That's not it. Okay, so basically what I'm trying to do right now is just figure out where exactly the gene starts. Because if I grab this and I hit create translation forward, delete this other one. So if I grab the GFP, you'll see that all of... So this is the amino acid. So like DNA is broken into 3-letter codons. So it's like these 3-letter chunks, and each 3-letter codes for one amino acid. And it always starts with an ATG. ATG is like start. It also codes for an amino acid called methionine, which is this M here. So you'll see that if I grab the GFP here, and I click make translation. So basically this is like read the DNA, tell me what amino acids it's going to be. You'll see that it's a nice continuous thing the whole way through. There's no stop codon until presumably way at the end. In fact, if I just do this and I go create translation forward, yeah, it's right here. So the little star is what's called a stop codon. So it literally means like stop. Like this is as the DNA is being read, or well, really it would be the RNA, but that's neither here nor there. As this is being read and turned into protein, when it hits these little stars, the thing stops. It's like this is the end of the protein. But the thing is, what's throwing me off here is there's these other things like this his tag. So if I grab it and I hit forward, it makes histidines, which is another amino acid. And the reason that you would put a his tag on something is because histidines stick to nickel. So you can basically grow your protein in the organism that you're interested in and then run it through a column with nickel in it, and only the protein you're interested in will stick to the column and everything else will just run right through. And then you can add another compound and make it let go of the nickel, and then you can collect your product. So this is a really common thing to do for, say, how they make insulin industrially. They'll put a his tag on the insulin, so that way that they can grow the insulin in a tank, usually in E. coli, and then extract it via this his tag. So when I see a his tag, I'm almost always a little bit suspicious that the start of the protein must be before this inherently, because otherwise this wouldn't get stuck to the like if this is just here and there's a stop codon here, these two things will not mesh, right? So if I grab this from the beginning, which is here, and I go to here and go make translation, you'll see, and this is what was worrying me, every single one of these is what's called in frame. Actually, is there a gap there? There is a gap. Huh. That's really weird. That's really, really weird. There's actually a gap here. Huh. Okay. So the reason this is now throwing me is because there's this one letter gap here. So a one-letter gap in DNA code will absolutely fuck this entire protein, because, and I can show you why in a second. So like let's pretend that, so if this letter wasn't here, it's a G in this case. If this wasn't here, then this his tag would be what's called in frame with the GFP. So if you were to start transcribing this protein, and start making the let's pretend that it starts here for a second. So it's making protein, it would just keep going all the way through this GFP. But here's the problem. Oh, somebody just chipped in. Thank you very much. Thank you, El Sylvia. But now let's grab from this missing letter, and we're going to just tell it to make a little bit of a translation, because I assume this is going to get fucked almost instantly. If we go forward, do you see what happens immediately? Like literally within 2 amino acids, there's a stop codon. This is super bad. This is like super, super, super duper bad. Because what would happen is this entire GFP just wouldn't get made. Not only would it be making the wrong amino acid, so it would just make junk, there's these stop codons here which would completely ruin any GFP from getting produced. So this is very weird. This makes me think that the start must be somewhere in here, and this his tag is maybe left over from some other bit of, like, while they were building this plasmid, like that. I think this his tag must be left over from that, because there's no way that this GFP would work if this his tag was there, which I don't know. It's very strange. It's very, very, very strange. So I'm not entirely sure what to do about this. I'm going to delete all these translations because they're just kind of clogging this up at this point. I'm definitely going to delete this one. So this one's important. We want to keep that. This one we don't need. Going to get rid of that. So I don't know what this is for. I don't see a promoter in... Well, so I see a promoter here. At least I assume that's what this is. But it doesn't... I don't know. Something about this is just... Like... You know, my spider sense is tingling kind of thing. Like, it's the Peter tingle. Something about this is throwing me as like, something doesn't look quite right, and I can't quite place what it is. So what I'm thinking is we're going to try and keep this as intact as physically possible. So if we were to say use this BAM-HI site, this is a restriction site. So if I mix BAM-HI, which is the name of an enzyme, with this piece of DNA, it would cut this piece of DNA right here. So you can see how it, when I highlight it, it like highlights how it cuts. So in this case, it cuts from so right between these two G's, And then it leaves a bit of a dangle. So it's this GATC is the dangly bit here. So it would go like zoop, boop, boop. So this is what's called a sticky end. So this is particularly helpful when you're using what's called Golden Gate assembly. So you're using restriction enzymes primarily to like cut things apart, which is not super convenient because it means that you essentially have to just work with what things are here. So what we're going to do is we're going to stick the actual proteins that we're interested in this area, but we're going to make sure that whatever this thing is still there, and we're going to make sure that it remains in frame with this GFP. So we're going to assume that this works, because the thing is, I have this in my fridge, but I've never actually gotten it to work. Like, I've grown it before, but I never got any GFP production. So it could mean this was assembled improperly, and that weird one-letter gap right here actually is lethal and destroys this, and is the reason why I couldn't get any GFP, or I just wasn't using the right light. So I'm not really sure. I'm going to end up tinkering with that on my own time, but to use this as an actual like base for gene engineering, we're going to just pretend that this is done properly, because if it isn't, it's very easy to just, bip a letter and just hope for the best. So what we're going to do is we're going to cut with, inevitably, we're going to cut with BAM-HI, but we're going to make sure that we put this back, and we're going to make sure that whatever we put in here is going to have, essentially what we're going to do is we're going to just include this little sequence of probably junk, and then make sure that whatever we stick in here starts at the same spot where this GFP would start. Because that way we can guarantee that it is at the very least going to behave exactly the way that this baseline plasmid would, and we're not going to get any we wouldn't expect any weirdness from it, being frame shifted or something. But if there's a frame shift in here and whoever made this is just a doofus and didn't catch it, then we'd have to figure that out later. But we're going to just assume that they did it correctly, because I always want to assume that people aren't idiots, because, that's just polite. And so then the other restriction enzyme that we're going to use, because essentially what we've got to do is cut this whole chunk out, because it's basically useless. Also, the other reason why I don't necessarily think that those his tag, like the his tag at the front, that I was just worried about, is real, and why I think it's just a remnant from like the assembly of this thing, is because at the end of the protein there's these two stop codons, and they're before the his tag. So why would you put this in? Why would you have this his tag after a stop codon? That would literally, this will never get made. So it's very strange that this is here, but stop codons are here, which makes me think that this is left over. Like, this is not actually functional, which makes me think that this too is also not functional and is just left over from however the hell they built this plasmid. Okay. Okay. With that all said, we are now officially ready to actually get working. So, like I said, because we're going to keep this whole section, what I'm going to do is I'm going to just literally grab the GFP and just delete it. And we're going to just assume that anything between this XHOI and the BAM-HI here is going to include whatever junk is not, basically these stop codons all the way to here. We're gonna assume that this would have to be included in whatever piece of DNA you get printed, and we're going to assume that whatever this is also going to get printed with whatever you end up making. Thank you, El Sylvia. Greatly appreciate it. So that way we can essentially know that whatever we have printed should theoretically work. And I keep saying what we get printed as if we're ever going to actually make this. There's no reason to make this. This is really just proof of concept because I think it'd be fun. But I don't necessarily think that we would actually have this made, just to be clear. So yeah. This is nquois bar 1 x by. I'm also trying to see if there's a way to take the. Actually, does Adgene have pdusk? That would be really helpful if they did. Oh, it's here. They do have pdusk, or they have something that looks like pdusk. P-dusk. There we go. Empty backbone. Wonderful. So again, this looks like it's got the same weird issue of they did not label their shit, which is annoying. But this is P-dusk. So what we could do is we could take this chunk thing and use this as our dark trigger, and then we can take this bit and use it as our light trigger, and in theory you could sort of shmoo them into one plasmid. The problem here is they did not label their shit, so it's like there's no information here, and it's very annoying. I hate when people do this. It is extremely frustrating. If you ever make DNA, fucking label it. Like, don't make me come over there and beat you over the head with a pipette. Like, fucking label your DNA, because otherwise it's useless. Like, how am I supposed to look at a bunch of random-ass letters and know what the hell I'm looking at? I'm not. It's impossible. Like, I could run BLAST and then do, try and piece it together, but that's a huge amount of work. Or you could just put a label on it. Ugh. Anyway. Okay. So we're going to come back to the P-dusk. Let's pick this first. So, big question. What do we put in here? So, I mean, we can actually leave the GFP in here. There's no reason that we'd have to get rid of it necessarily. Like, we could just have leave it in there as a tag. In fact, I think that's probably what we will do. So we have a few different options. We could have this make a flavor, or we could have this make a texture, or we could have it make both. So, like, for example, we could, in the last episode, which I will open up the thing that we made last time, berry cheese. So this is Cheeseberry McParaface, which is our super elegantly named, and you bet your ass if I ever submit this to AdGene, I'm fucking calling it that, because I'm an adult and I can do what I like, and professionalism is for people with artistic integrity, and artistic integrity is for squares. So this is what we made in the last episode. So it's got a whole bunch of different genes in here. So the important ones are ATF1 and linalool synthase. So linalool, which is what linalool synthase makes, is known as the sort of Fruit Loop chemical. So it smells reminiscent of Fruit Loops. ATF1 is Acetyl Transferase. So what this one does is it takes alcohols, like long chain alcohols, or not like ethanol, and more think like hexanol, and it sticks an acetyl group, like it makes an ester and it sticks an acetyl group to it. In fact, I'll just show you what this one looks like. Yeah, there we go. This is the graphic I'm looking for. Okay, so here, this is what ATF1 does. It takes these long chain alcohols and it makes an ester and it sticks an acetyl group to it. So Cheeseberry McParaface included all of the compounds that massively upregulate the production of this alcohol, hexanol. So when hexanol is acted on by ATF1, it produces hexyl acetate, which smells like pears. Hence Cheeseberry McParaface. Because it's a mix of pear scent, Fruit Loop scent, and it works in lactobacillus. So it would work in a species of bacteria that is used to make yogurt or cheese. So hence Cheeseberry McParaface. So we could include linalool synthase. So we could grab this, and we could put that into P-dusk, or Pdon, and so like when you expose it to light, it should smell like Fruit Loops. That sounds pretty cool. So we could do that. Or we could grab one of the proteins from, say, our egg or milk. Where's the other one? Our milk plasmids. So we could have it be, make sort of like a cheesy texture, or like a goopy egg white texture when exposed to light, and that could be kind of cool. So it's kind of I'm gonna kind of leave it up to you guys. I'm seeing the Doctor Who memes already. Ugh, a pear. I hate pears. Too mushy. Um, anyways, so yeah, what do you guys think? What should we have the light trigger? Should it be a Fruit Loop scent? Should it be a pear scent? Should it be a texture? Should it be both? What do you guys think? Vivek shooting pipette tips at each other. I never really worked in a graduate lab or anything like that, so I never had that opportunity. But that sounds like exactly the sort of thing people would do. It's hilarious. Sour in the dark or sweet in the light. That could be interesting. Bill Rubin. Why does that sound so familiar? Rubin? What is this? Is a yellow compound. Why would you want this? This is like piss color. Gross. Um, Spicy and light. Spicy would be hard. Spicy is a huge pathway. Um, Spicy would be fun. I mean, so like this is one of those sort of like, the world is your oyster, but the chance of it working is slim. So yes, theoretically you could make spicy or vanilla or whatever else or mint. So also don't think that we only have linalool as an option here. So like things that are probably reasonable. Limonene would be fine. So like a lemon. Linalool, ATF1, and we could throw in the like hexanol or octanol pathway. Although I have hexanol, so I'd prefer to use that, so I don't have to do a whole freaking pathway again because that's annoying. Um, but also think that we have some texture modifiers as well. So the caseins, the lactobumins, ovalbumin. Um, I don't really know what else we could do for texture. But yeah, so what do you guys think? Bitter would be hard. Bitter is usually a really complicated chemical, so it's a whole pathway, and I'm not familiar with them enough to know what's a good like bitter compound. Four hydroxyphenyl butane. I swear to God, if this is drugs, you're getting banned. Just so you know. Raspberry ketone. Yeah, we looked at this one last time, and it's not easy to make synthetically. It's like it's a huge pathway. Um, all right. So what's the consensus, guys? What do we want to make? Um, I'm getting some sense. I'm getting milk and eggs. Come on, pick. Yeah, denatonium. You know. Not only is I don't know if you can make a denatonium benzoate synthetically, it would be extremely hard. Um, lemon. Okay, let's go with limonene. Wait, I've seen this on Adgene. Let's see if we can find this. Limonene synthase. Bacterial. There we go. That's easy. Not frickin' labeled, but, you know. So I actually have one of these. Come on, Sylvia. No, don't be a fuck, please. Oh, thank you, Simon. Greatly appreciated. Oh, quinine. How is quinine made? That could be interesting. Biosynthesis. Oh, Jesus. Okay, yeah, no. Hard no. Yeah. No. I like the idea, but that's some really hard biochemistry. That's a lot of it. That's 1, 2, 3, 4, 5, 6, 7, 8, 9 enzymes, of which, and using a precursor which doesn't exist in bacteria. That'll never work. Apple lemon is nice. I don't know if I can do. I don't know if you can do apple. I can do lemon. I can do lemon. Lac Repressor. Lac operator. So yeah, it's got to be right here. I don't know. Yeah, screw it. Let's download this. Limonene synthase sounds fun. No, that's not what I wanted. I just wanted to click on this. Shit. Okay, so I'm actually leaning towards lemon, because lemon sounds fun. Just download the gene bank file, and pop it into the old taste goo folder. Okay. Import to. Should be this guy. Rename this, because it's always important to label your shit. Cool. So the limonene synthase should be in here somewhere. So let's see if we can find it. There's the his tag. It says the his tag was stuck to it. So, okay. This is a ribosome binding site, which is where the actual ribosome will stick. Ribosome binding site is almost immediately before the start of transcription. I see an ATG right here. I do not see any others before that. So we're going to go ahead and assume that it goes like this until we see a terminator, which is right here. Oh, no, that's another his tag. Okay. So let's just do this. Let's go till like here, and then let's click make translation and see if it makes a bunch of junk or if it actually makes the thing that we want. And by the looks of it, it makes the thing that we want. So you'll see there's absolutely no stop codons, which to me screams this is the real protein. And it actually does start here. So that's cool. I don't care about this his tag, so I'm going to ignore it. And I'm going to go from there until I hit a stop codon, which I saw at the end there. Yeah, it's right here. So we're going to just go till right there. Make sure that I'm not missing a letter at the top, which I am not. Copy. Cool. And now we go over to our Codon GFP, which I'm going to actually duplicate just to make sure that I have a blank copy in case I fuck something up. Yeah, lemon and light beer does sound possible and does sound doable. So I have a plasmid that is supposed to do limonene synthase in yeast. The only problem is, the only way to get it into the yeast is to use dropout media, and I think I've talked about this in a previous video, or at the very least a previous stream. Yeast are able to produce most of the amino acids themselves, but there's, I think, 5 that they can't. So normally when you grow yeast, like even if you're growing beer or whatever, even if you don't realize it, you're providing those extra amino acids via something that you've added to the growing media. Be that through the fruit juice that you add if you're making wine, or the mash that you add if you're making beer, or any of that kind of stuff. You always have to provide those amino acids. So One way of genetically modifying yeast is to include a gene in the plasmid that you want the yeast to take in that allows them to synthesize one of those missing amino acids. The problem with this is that means you need media that contains everything other than that one missing amino acid. This is very expensive. It's like $300 for the amino acid mix, and then like another 150 potentially for the nitrogen base, and then a carbon base. It's a fucking nightmare. It's very expensive. It's very annoying, but it means no antibiotic selection. So the that plasmid uses, I don't actually remember. I don't remember I mean, fuck it, I can check. Actually, yeah, I have this open, so this will take like 2 seconds to find, potentially. I got it off of Adgene, so yeah, it's this one. So this is the one that I have. It uses, yeah, leucine. So the limonene synthase is supposed to be in here somewhere, although I've run BLAST on this and I couldn't actually find it, so it's in there in theory. But it uses leucine dropout media, which I don't have. I just noticed it's got a 2 micron Ori though. That's interesting. But yeah, so it uses leucine dropout, which I don't have. It's very expensive. I don't feel like buying it. So just to be able to use this plasmid, I would essentially have to cut it open, stick a G418 resistance gene into it, and then be able to use it. But then, yes, you could make yeast that tastes like lemons. And limonene, I believe, is a small enough compound that it'll actually exit the yeast. Unlike my beta-carotene, which is frickin' massive, and refuses to leave the yeast, which is actually really annoying because I was trying to make like beta-carotene beer, but it's not happening. Well, I mean, it's working. Like the yeast are growing and it's making wine. It's just the carotene isn't going into solution like I wanted it to. I'd have to physically pop the yeast open to get it to do that. But you could using one of the systems that we were looking at the very beginning of the stream, where it's kind of like seeing if we could do this in yeast, you could do limonene synthase. So you could take this limonene gene and stick it into a light-triggered yeast plasmid, and then put it into yeast, and then yes, you could do that thing. So okay, we're gonna go back to the Pedon. We're gonna go back over to here. I'm gonna leave all the GFP there, and I'm gonna just go pasty paste. And I'm going to create annotation, and I'm going to call it limonene synthase. So, and I'm going to give it a color. Of course, I'm putting it yellow because, duh. Let's close my annotations. And now we can see that I've stuck limonene synthase next to the GFP. So technically, when this runs, it should make these bacteria smell like lemons and also turn green, which I think is dope. So technically, we could leave it here. But come on, where's this? Okay. So I actually have to add a start codon here. So I'm going to just go ATG, click add. So this is actually where this starts, just because gotta have a start codon or it doesn't work. So right now we've got a limonene synthase GFP light triggered thing. So of course this is in E. coli, so you really wouldn't want to eat this, but it's like I said at the beginning, it's really just a proof of concept. Once you had this working, you could then work on essentially porting it over to a food-safe microbe, so something like a yeast or something like a lactobacillus or something like that. Oh, a little update on the antiviral therapy for plants. It looks like my plants don't actually have a virus. It looks like it was actually just insect damage. And thanks to that video, people wrote in and they're like, yeah, really, that looks like insect damage. And as the plants have continued to grow, and I treated the plants assuming that it was insect damage, it looks like the damage has gone away. So all the new leaves show no damage. They look healthy. They're growing perfectly fine. So it doesn't look like I actually even need to do the antiviral thing. And also, one of the like key reagents to being able to do the antiviral thing for plants, I don't actually have. So it literally is impossible for me to do it. So yeah, that idea, up in flames. Kind of annoying, but actually kind of good, because it means I don't need to do the thing because my plants aren't infected, which frankly is better. Cool. So now we've got our, in light, it becomes lemon. Now it's kind of a question of, do you do... I mean, because one thing that we could do... did I import that yet? Did I download that? Bedusk. Here we go. Download. So let's load this in. So let's make a different version of this now. So this is Pdusk. So this is the one that works in darkness. So what we could do... Where is it? Oh, here. Rename Pdusk. Cool. So assuming that this is using the same thing as before, here's this BAM-HI site. This was what was present in the... I mean, there's clearly a multiple cloning site here. Like, it doesn't make any sense that they would put this multiple cloning site here and not use it. But ROP, I don't know what this is. Bomb, I don't know what this is. Lac Promoter. Okay, we're gonna just assume that this is the correct site because I don't know. I don't have any better ideas. So I mean, in the PDON thing, at the very top there was BAM-HI, and at the very bottom there was XHOI, right here. So it makes me think that, that's... I mean, it looks the same, right? Like Pdusk and PDON are almost identical. So if we scroll up a little bit, you see the F1 Ori is right here. So it makes sense that between here and here, in these same two highlighted restriction sites, it makes sense that this is where we could put our gene of interest. And so we're going to just operate under the assumption that that's correct, because I don't have any better information. I'm not necessarily going to use XY, because that's actually really bad practice to cut out this entire multiple cloning site when these enzymes could be very useful, and then it would leave us room to like keep working on this. I fucking hate when people put lazy restriction sites on their plasmids. It's annoying. Or rather, when they cut out all the useful ones and then they don't leave you stuff to like continue to work on the plasmid. Like, it's just bad practice. So one thing that we could do, so you see how this says CANR? If we were to add like a different selection marker, I mean, so we basically have two options here. We could either, I mean, the promoter would be different. I don't know. So like, there's one option where what we could do is basically cut out. So yeah, it's the Terminator here. So if we cut out, this is the F1 Ori. If we cut this stuff, basically all the way to essentially here, should hit this, which is that. Okay. So theoretically, what you could do is if you were to like cut from like here, nope, other way, to here, and like stick this whole thing into this plasmid, just because the problem with this is it's all poorly labeled. So like I don't know where the correct parts I'm supposed to like use are. Strong lambda repressor. Yeah, so it's like if you were to basically combine these two, you could do that. The other way to do this is if you could sub this out. So like if you take this kanamycin resistance gene, which is that's what CANR stands for. If you were to replace this with like ampicillin or something, like, or tetracycline, or just some other resistance gene, what you could do is you could co-transform both plasmids into the same bacteria at the same time. So basically, as long as, like, if you made plates, so like petri dishes, that contained both kanamycin and whatever other antibiotic you want that you would replace this from. Only bacteria that took in both plasmids would be able to survive on the plates. And now this isn't super great practice because now you're essentially making bacteria that are resistant to very different antibiotics, and can't necessarily suggest that, but just for the sake of argument here, that way you wouldn't have to try and combine both plasmids. You could just kind of co-transform them, which admittedly getting two plasmids into one bacteria is a pain in the ass, but it is doable. And then that way you could have it so that under dark conditions it does one thing, and then under light conditions it does another thing. Not really sure. Yeah, not really sure the best way to go about this. I honestly don't know how to combine these because this is so poorly labeled, and it's really, really frustrating. So this is actually a really great excuse to talk about label your shit. Like, the fact that there's just this, like, chasm of empty God knows what's in here makes this really, really frustrating to work with. And I fucking hate when people do this because it's annoying. Like, it makes it hard to know, what bits can you touch, which parts can you not. Then you got to like go through this manually and try and figure out like, all right, well, what's over here, and it's just frustrating. It's a giant pain in the ass when it's like the person who made this could have just labeled their shit. And what's really funny is this was made by professionals and they don't know how to label their shit, which is like, y'all need to work in a kitchen or something, because if you don't label your shit in a kitchen, you get thrown the fuck out. And like, it's an important skill. So, I think we might actually be a little bit shit out of luck. I'm going to make a PDusk that does something else, and we're going to just pretend that you could sub this out for something else. But until I actually, take the time to, like, go through all of this. Excuse me. Fuck. And actually figure out, like, where the various parts of the PDusk thing in here are, it becomes essentially, like, useless. So let's just go back over to here. So we did lemon on the other one. So I don't see a start codon. So I think we can just add one in. So we're going to say, yeah, I don't see any ATG. Yeah, there's no start codon here, so we can just add one. So I'm going to use EcoRI rather than BAMHI because I'm just trying to teach good practice here. Always leave an extra restriction site. Always, 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 always. Like, it's just good practice. So we're going to go EcoRI to Hind 3, I think. So this is going to go, and it's going to be replaced by whatever we feel like sticking in here. So speaking of which, what do we feel like sticking in here? Okay, so we have a question here. Why can't you just build a plasmid from scratch? Is that significantly harder? Harder? No. More expensive? Yes. So I actually just have been looking at getting a plasmid made entirely from scratch, and I'm waiting on a quote, but I'm expecting it to be... So if you start with a backbone, like where most of it's already made, you're basically... The thing that you're paying for, essentially, is the thing that you're sticking in here to be synthesized. Usually. So in this case, what you're paying for is essentially a fragment of length 1,665. So by my rough guesstimation, that would cost you in the range of $500 to $600 to get this one little piece made and then put into this backbone. So you tell the company, cut here, and cut here, stick this in, call it a day. Because we left the GFP in here, you'd actually have to resynthesize that GFP as well, so it's a little longer. At $2,300, that would put you into the $800 or $900 range. Whereas if you're trying to synthesize this entire plasmid from scratch, that's 9,000 base pairs. So that becomes much more expensive. That is in the range of $2,500 to $3,500 to do this from scratch. Which, if you've got grant funding, that's basically chump change. But for people who are trying to do this from more of like a DIY or independent researcher type thing, or even if you're just working in a lab and you don't have infinite grant money, like starting from a backbone is important. It saves you time theoretically, it saves you work theoretically, and it saves you money theoretically. Because like buying this backbone costs $75. So $75 plus another $500 for the thing that you want to stick in there is a lot less than having the entire thing made and having to pay $3 or $4 grand. So I'm actually going to be, especially since I've been doing these streams and because there's been a lot of interest, I'm actually going to be working on a video about like intro to genetic engineering, basically. And I mean, technically I have already made that video. In fact, I've made that video many times. Like I've done lots of things that are sort of intro to genetic engineering, but the thing that I've got planned is going to be it's going to be kind of more stripped down. It's going to be more having like links to different resources, covering the basics, all that kind of stuff. So yeah, I'll do that eventually. But yeah, again, the manga guide, excuse me, the manga guide to molecular biology is the thing that I always suggest. It's a great resource. Okay, so let's see if we can just wrap this up because I'm getting kind of tired, and I gotta go get more dirt. I got a lot. Like I was late to the stream today because I was getting plants, and I need to go buy more dirt. Okay, so what do we want to stick here? I don't know. I'm thinking let's just do linalool. Let's have it do Fruit Loops in the dark and lemon in the light. That sounds pretty interesting. Yeah. Also, I don't sell kits. That's not my thing. That ain't me. Okay, so we're gonna go. Reverse complement. How do I? Translation, reverse translation. No, that's not right. RNA sequence. Oh, somebody kicked in. Oh, thank you, Spectral Bacon. Okay, so I don't know how I'm going to actually do this, because this is pointing in the wrong direction. So I got to figure out how to do a little flipperoo. Okay, this is highlighted, right? So if I just grab... Well, actually, we were going to do linalool, right? Somebody... Oh, thank you, Nico. Okay, we're going to do linalool synthase, because why not? Text reverser. I need this pointing in the other direction, because it is ass backwards. This, I think. Yeah, that's what we want. Cool. Yeah, it's funny that we're essentially using word processing tools to do this, because, I mean, that's basically what we're doing. Okay, so we're going to go to PDusk. The only reason I had to do the little reverse there is because this is pointing literally in the wrong direction. So rather than going and messing with these restriction sites, I'm basically going to... So this goes from the G, and this goes from this T. So we're going to... Oh, okay, my mouse decided to take a vacation. I believe that goes to this G. Yes, cool. So we're going to delete that, and we're going to paste in our thing. And we're going to create an annotation. And we grabbed linalool synthase, so linalool synthase, strand, I mean, it's just pointing in the other direction. Wait, shit, do I need the complement? That's a problem. Shit. It's okay. So not only do I need this on the I needed to reverse it, but I also needed the reverse complement. Shit. Okay, wait. So I can fix this. I can fix this. We want the reverse complement. Okay. Yeah. So we're going to copy this. We're going to then delete this. Delete. And then we're going to paste the reverse complement in here. And that should have fixed it. Because the ATG should now be on the bottom. I'm an idiot. Okay. I was reading the wrong line. Okay, wait. I'm going to undo some stuff. I need to just check something. I have the right thing copied now, so I should be able to unfuck this. Okay, yeah. I was reading the wrong line. Okay, so when you're going, if you're going clockwise on a plasmid, you're reading the top strand, but if you're going counterclockwise, you got to read the other strand. And I forgot this, and now I feel really stupid because I should not have forgotten that. I'm supposed to be a professional. So I was looking for ATGs, but I was looking on the wrong line. So I don't know if there's any in here. I don't think there is, but just in case, I needed to undo everything so that I could actually check. The nice thing is I can still just redo the thing. Thank you, El Silva. I can just redo this really quickly. I don't see any ATGs, so we're going to just grab this and go to here. That leaves that. Okay, we're going to delete that again. We're going to paste this again because that should still be saved. That's still labeled. That doesn't look right. That doesn't look right at all. No, this is not right. I fucked something up. Shit. It's very frustrating. Somebody else put you in? No, it was also you. Okay, cool. Butts. Okay. This is extremely frustrating. I grabbed the wrong thing, and now I don't know how to fix it. So you can see that, like, this is going clockwise. I need to make this counterclockwise, and I don't know how to fix that. So this is the DNA sequence. Why does the reverse complement look wrong? TAC. Okay, so this is that. So, like, I need this backwards, and I also need, I mean, what if I copied this? What if I copy this, go over to here, heat that, paste this, grab this. No, because if I paste that in, it's just going to give me the same fucking thing in the same order. Ow. Oh, no, wait. If I copy the reverse complement, maybe... What if I just paste that in? What if I'm overthinking this? Which I suspect I am. Okay, we go back to ptask. Click, click. Paste. It worked! Yes! Okay. I was overthinking this. The program was actually doing the thing that I wanted it to. I just... Yeah. I was just overthinking it. Wait. No. It needs to be... CTA. Because we're doing on the reverse. Okay, and now if I go make reverse translation, that should be a stop codon. And it is. Wonderful. Man, that is a mind fuck to have to work backwards and upside down. But I do not recommend at all. There's a weird terminator there. It's going the wrong way. Don't know what to do about it. Don't care. So yeah, we're gonna say that this is our this would make Fruit Loops in darkness, and then this one would make... I need to rename this. Pinon limonene GFP. So yeah, this one. So the first thing that we made, which is this guy, makes lemon scent in light, and then this one, which we just made, in theory, assuming I didn't fuck something up, which I almost definitely did, would make Fruit Loop in darkness. Yeah, so I think that's a good stopping point. I'm gonna take a few minutes to answer some questions, but I think that's a good stopping point for today. I wanna dig more into this in a future episode. I'm gonna take some more time off camera to read up on those yeast systems, and see if I can find a good collection of plasmids that are like a good baseline, so that way we actually have something to work from. I think I just needed to do a little more prep for this. But I mean, this is a good, like, dip into the water, so to speak. So we'll come back to this light activated flavor thing. I like this concept. I really do. I really would like to see if we can make it in something that's actually like food. Just because I think it'd be neat. It's just, I find it to be a really cool idea, and it's something we could build on. I'd love to make a plasmid that has like a proper multiple cloning site and is properly labeled and all that kind of stuff, so that it's easier to do this sort of thing. But as like a good, proof of concept, having one that does lemon and one that does Fruit Loops, and having it be able to like change based on light levels, I think is really cool. So I think, yeah, that's where we're gonna end it for this design session. I'm going to take a little bit of time to answer some questions, and then we're going to call it for the day. And because I need to go to the store and buy more dirt so that my plants don't get really, really sad. Okay, let's see. What do we got in the old question department? So Joe asks, what is the simplest life form that you can possibly make? And now I don't remember the exact name for this, but I can look this up because I know where to find it. Free Genes. So Free Genes has made 3 things thus far. And where do you order this? I want to find this because somebody has actually, there's been a group who's been working on making like the minimal like a minimal cell, and I don't remember what it's called. I want to say it's the JVC collection, but yeah, or JCVI, that's what it's called. JCVI V3 bacteria. Yeah, so it's let's see if I can find it. Yeah, this is it. So basically what they did was they made, I mean, this is actually a better graphic. So essentially what they did was they synthesized all these individual little pieces of DNA, and then they built them up into progressively larger pieces of DNA that eventually turned into one gigantic, I say gigantic, it's freaking tiny, super duper stripped down entire synthetic genome. So it's an entire bacteria with as few letters as physically possible. I think it's 1 million base pairs, which sounds like a lot, but like it really isn't. And I mean, they've been working on this to, so now they're, I think, down to, I think it might be down to 500,000. They've been slowly but surely, like, removing as many parts of the genome as physically possible to see at what point the thing breaks. But this is totally like a thing. It's super tiny, like teeny tiny. So basically what they did was they started with the smallest known bacteria, like with the smallest known genome, and they just started removing things to see at what point it breaks. And they kept removing until they basically hit where they're at now. So they're on Syn3, which is Mycoplasma Laboratorium, which is, this is what I mean. Fucking, like, biologists are nerds. So yeah, they removed 482 genes out of the original genome, and they were down to 382, which sounds like a lot, but like considering it's a living organism and it only has 382 genes, that's crazy. can we stop comparing people to Elon Musk? Like, Elon Musk's greatest accomplishment is having shitloads of money and like being good at rewriting history. The dude's not actually like let's maybe get off the altar of Musk, please. It's really frustrating. Also, he's a bit of an asshole. I'll happily say that. Like, said a lot of things that are really awful, and he's currently trying to force his workers back into Tesla plants in unsafe conditions. So yeah, let's get off the frickin' altar of Musk, please. Okay, so given your opinion of university being too theoretical, what's your opinion on middle high school? Okay, fucking do high school, like, and even university. It's like, okay, so let's be really clear here. School is not perfect. That's a given. But it does serve a job, and some people are. I will answer milk and eggs in a second. Some people are much better at learning in a school environment than they are on their own. Like, learning on your own takes a specific type of discipline, and it takes a specific type of person to be able to keep at it and get your shit done and actually like learn the thing. Also, school comes with magical piece of paper at the end of it that says, like, we guarantee that this person is not a fuckwit. That's basically what school is for. If you're a weird case like me, I've been able to, through a variety of situations, have been able to build myself up a profile and a portfolio, rather, that can prove that same thing, that I am not a fuckwit. But school has that purpose. Doing middle school and high school, I think most middle school and high school curriculum in most places is kind of shit. I think there's a much better way that they could teach it. I think there's much more information that they could convey and a lot less crap that they could convey. But to say that the entire system should just be thrown out is not fair, and it serves a purpose. There are definitely things, and also, like, the point of school is not necessarily the things you're learning in class. The things you learn in class will mostly be forgotten. That's a given. Unless you're, someone like myself who has a reasonable memory and fucking paid attention. But, like, most people forget the majority of things they learned in school. Like, you're learning calculus in high school. Like, most people end up abusing it. But what high school is really good for is first of all, it forces you to be in social situations. So it forces you to, build a social life and also social skills, which is super important. And frankly, more important than most of the things you end up learning anyway. It tries to help you build habits that are important for the rest of your life. And it's the same with university. University, in theory, is supposed to teach you all of these things. And, more importantly, it's an excuse to be with peers of your own age so that you can, and slightly older, so that you can get and also be exposed to people who are much older than you, who can give you different perspectives than what you may already have, and give you different experience than what you may already have. And in theory, the labs and such should teach you how to do the thing. It's just, you got to try and find a program that will teach you the things that you want, and you've got to, try and find a university that will teach you the things that you want and has the facilities that you want, and it's tricky. But even, like, a community college science education is better than nothing in a lot of cases. Unless you're a bit of a nutsack like I am, and, like, a nutcase who just can learn on their own really well, school's important. Like, it does the thing. So yeah, don't be skipping high school because you think you can, like, learn on your own. Get your freaking diploma or whatever, like, and also, like, university isn't necessarily for everyone. Like, college and, like, apprenticeships and trades tend to be downplayed, but, like, they're super important. They make really good money, and can be much more fulfilling than a lot of, like, science degrees. Like, science degrees, you end up just getting fisted for like 12 years. Like, that's most of what a science degree is. It's, you do your 4 years of undergrad, and then you do a master's, and then you do a PhD, where you're mostly underappreciated and underpaid the entire time. And by the end of it, you walk away with a pile of debt and, a minimum of respect from people around you. Whereas, you go be a plumber, you walk out of your apprenticeship making money, like, being in, like having money, not being in debt, and having a job which is always going to be necessary, and you can a lot of the time set your own hours and do all kinds of stuff and get a job right out of school. There are different options for different people. Don't think that one thing is the perfect solution for everybody, Everybody's different. Everybody is able to do things differently. But, that's not to say that school doesn't have a place. School totally has a place, just not for everybody. And also, I know a lot of people in the trades who, they do that for work, but it's also given them the skills to go off and do crazy, crazy things. I mean, look at, like, Colin Furze. Dude was a plumber, and now he makes fucking, like, hover bikes and, like, Wolverine claws for a living. Obviously, that is the exception, not the rule. But, doing a trade is a great way to make money. It's a great way to, like, learn some real-world skills. Will it make you no one's going to call you a doctor afterwards, but it's a way of getting through life and doing a thing, and also then giving you the time to do things like genetic engineering on your spare time. So, like, if you want to take the I want to learn on my own route, you also got to figure out, but how are you going to make money throughout that whole time, because you need to be able to eat. So, until we have UBI, which is, thanks to the pandemic, we might get there sooner than rather than later. It's a thing. So yeah. Okay, what else we got in things? But yeah, kids, stay in school, don't do drugs. Okay, being a microbiologist, where would you point to start learning more about genetic engineering and coding for biological engineering? Waldo has been saying this for me. The manga guide to molecular biology is a great starting resource. From there, you're getting into much harder, like, info-dense texts and that sort of thing. But, reading up on all the information on the BioBrick wiki, picking up a genetics handbook, or not handbook, textbook, and then, but really from there, it's just kind of like pick a thing and just start reading up on how it works. Like read lots of papers, figure out how the pieces go together. But like there's some basics, like this is a restriction enzyme. This is this is ligase. This is how these things work. You just the open wetware protocols are really great because they'll explain how certain things work. But the manga guide is always a great starting point. Joe Bennett asked, why haven't you reapplied your lactase gene therapy? Because I am waiting until we have made the better version. Alright, what else we got? Can you use an air gun as a gene gun? Technically, but there are better ways of doing that thing. Okay, I can't pronounce her name. Mercedes? I just applied for the Bio University. Can you explain a bit how do the start and stop codons work? My books are like, they exist and they work like magic. Welcome to biology. That is most of it. We don't know why they work, they just do. I don't have a better answer for you than this. Most in biology, there's a huge amount of stuff that we know that it works. We have no idea why it works. And there have been dozens and dozens and dozens of PhDs potentially studying that one specific thing that you're interested in, and people are like, well, we definitely know that this particular amino acid bends in this enzyme when these two things bind. Don't know why that helps, but we know that it happens. And so, like, it's this kind of thing. Like, we're like, how does this enzyme make this thing glow? Dunno. Like, we know that when this bond breaks, light comes out, but we don't know why. So yeah, it's sort of a mixed bag of, sometimes you just have to be comfortable knowing that we don't know, and just knowing that it works, and let that be enough. Can we have a protoplast fusion class, please? Yeah, just as soon as I get it working. Milk and eggs. Did it ever get so Milk and Eggs asks, did it ever get made, and is a stab possible? Milk and Eggs, it is officially getting made. It's fair warning. It's going to the milk and egg plasmids are going to take probably a couple of months to get made. The company is running basically a skeleton crew because there's a pandemic. But I just got the quote the other day. The order will officially go in. I mean, like, the order's in. I just gotta, like, send them the starting material, essentially, that they're missing to be able to do the thing. So, you know, that'll take a couple of weeks. And then it's gonna take them several weeks to several months to actually make the plasmids, and then it's gonna take more time to ship it back to me. So, yes, Milk and Eggs is totally getting made. Also, Joey Garcia, thank you very much. Greatly appreciate it. All right, let's do one or two more questions, and then I'm going to call it for the day because I'm tired. Can you gene edit eye color? I don't know. How bad do you want to go blind? Most of these questions are yes, technically, but messing with genes is a really good way to end up with horrible, horrible side effects. So, yeah, technically, probably. There is one gene you could turn off, but it is hard. There's also a laser eye therapy. I say therapy. It's like plastic laser surgery, plastic surgery laser treatment thing, where they can essentially like bleach your. And this is going to sound horrible. They essentially like bleach the pigment out of your eye so it goes blue. So, you know, that can be done without genetic engineering. And frankly, a laser into your eyeball is actually safer than attempting to genetically modify your eyes to be a different color. Okay, so if you can make these genes so quickly, why aren't there like crazy things on the market? End quote. The long and the short of this is, while it's easy to make these things, the regulation on their release is extremely stringent. So the amount of like red tape and stuff that you've got to like jump through and like hoops that you got to jump through to be able to release any of these things is extremely difficult. Especially in Canada, like to get something that's meant as food like approved, millions and millions and millions of dollars worth of investment needs to go into that just to pay for all the paperwork and lawyer fees and all the other crap and like the testing to prove that it's equivalent to the natural version. It's obnoxious, but I mean it's there to protect people in theory. I mean, fuck knows if it actually works, because then you end up with like a totally organic cucumber that's fucking, or not cucumber, like potatoes that are poisonous. Actually, I think it was a cucumber as well. Anyways, you can do totally natural breeding methods that end up with very, very poisonous food. And whereas the genetically modified stuff is perfectly safe, but they have to somehow prove that it's equivalent. It's the thing. So yeah. Ruben, how soon will we have perfect understanding of human genetics? Probably never. Yeah, that's the best answer I can give you. Probably never. Like, if you got every biologist into a room, like every genetic engineer and synthetic biologist and human biologist and doctor and everyone who studies the human genome and its interactions, and got them all in a room and was like, how much do we actually know? At best, 2%. We don't know shit. Like, we are wildly swinging in the dark, and we don't have a fucking clue what we're doing. All things considered. Like, we can do some very impressive things, but in the long run, we don't actually know shit. How do we get the chemicals to start transforming some bacteria? All you need is calcium chloride, and they sell it on Amazon. That's it, man. Like, you need calcium chloride. The rest are like reagents for handling the DNA and like cutting it apart and putting it back together. That you can get from NEB. You make an NEB account. Everything else you get from Thermo Fisher or Sigma. And so you basically make accounts with all of those places and you buy those things. Shouldn't there be finite problems at a given scale? I don't know what that means, but no. There's infinite problems. Like it's not code. It's like it looks like code. Like, it looks like we're coding because, like, look, these are letters. It looks like we know what we're doing, except it's not code. It's a physical system. This is an abstraction to make it easier to understand and process, but like, these are abstractions. Like, this makes a physical protein, which if you change one letter could fuck the entire protein from functioning properly. And like the only way you're gonna know if that one letter is important is if you change it and see if it breaks. So you're dealing with, I mean, this, what, how many amino acids is in this? Analyze translation. Here we go. There are, does it say? Or it's just a general count? It doesn't say. Okay. Wow, that's really big. So, I mean, if this is, actually, I can do this way. So this is 1,600, give or take, base pairs long, which divide that by 3. Fuck, I don't know. It's like 7 or 800. No, that's not right. A little more than 500 amino acids. That's 500 individual compounds that are all stuck together into a line that all have to come together in this weird, intricate dance of interacting with both water, the thing that it's dissolved in, and all of the other reagents and other chemicals and other proteins in one individual cell. Plus, you've got to account for all the individual cells interacting and their interactions with their medium and the environment, and it's so many things. Like, there is no supercomputer on Earth that could ever calculate even the most basic functions of a cell. Like, you could figure out, like, a protein, maybe, interacting with one compound, maybe, but it'll take huge amounts of compute time. It's extremely hard. Like, you cannot, like, just figure this shit out. It's too computationally expensive. It's very, very, very difficult. You're talking about the interactions of literally, like, quintillions of individual atoms. Actually, that's an understatement. It's like 10 to the 10 or 10 to the 20 individual atoms all like interacting in extremely complicated webs of interactions and protein interactions and chemical interactions. It's just extremely, extremely difficult. There is no simple way to just like reduce it down and solve. It doesn't work. Well, thank you, Nick. Greatly appreciate it. When you order your genes, will they mutate and go bad one day, or can they keep for years? No, they keep for years. They'll mutate once they're in an organism, potentially, but just pure DNA in a freezer, for the most part, is pretty stable. Some of the nitrogen bases can decay, and so you can end up with mutations, but for the most part, it's fairly stable. I mean, it depends how cold you keep it. If you keep it at minus 80 or like in liquid nitrogen, it can last for years and years and years and years. If you keep it in the fridge, it'll go bad very quickly, comparatively. Okay, I'm going to take one more and then I'm going to call it because I need food and other things. Yeah, maybe with some uninvented quantum computery magic. Sure, fine. But doesn't exist, won't exist for a very long time, and even if they did, they'd need a fucking massive one for it to ever actually be able to process anything reasonably. So yeah, it's still a long time off. Ever thought of doing any bioinformatics project? Not really. It's not really my thing. I know some people who are interested in it, but it's not my thing. I'm a nuts and bolts kind of guy. Like I don't really care how shit works. I just want it to work. So I'm more interested in like making things do interesting things rather than like trying to process huge data sets. It's just not my thing. Why do the mutations we insert into organisms disappear and natural genes don't? That is a misconception. They all get mixed and matched all the time. And this is gonna be my last question because this is gonna be a bit of a thing. Bacteria, especially like E. coli, which is the species that we end up using the most in the lab, they're mutating constantly. Like, some of them explicitly have DNA copying mechanisms that are... purpose is the wrong word because it's not purposeful, but like they have very lossy copying mechanisms, which they've kept not because they can't have something that copies their DNA more perfectly, but because the lossy mechanism is actually better. Like it makes more sense to have something be lossy and induce new mutations and give you the chance of gaining or losing bad functions or gaining new functions in various proteins and enzymes and whatever, than it is to just maintain the perfect DNA all the time. Especially ones that don't necessarily do like sexual reproduction. So there's no opportunity for their DNA to really get mixed and matched to prevent like lethal mutations from building up. Having a lossy copying mechanism when they're making like literally millions of copies of them, like the organism itself gets copied millions and millions and millions and millions of times over the span of a couple of hours. It makes more sense to have lossy copying mechanisms and have some of those copies be lethal, like, and have it just not, like, the organism grow and then immediately die, but also have the potential to have a beneficial mutation show up than it does to have a perfect copy every single time. Okay, so that we're gonna have to call it there because it's already 5 o'clock and I have other things to do today. I hope you guys have enjoyed it. This has been a lot of fun. We're going to come back to the optogenetics stuff in a future episode. This Friday, we're going to have another biohack chat with my buddy Gabe. So that's going to be a lot of fun. Not sure what the topic is. If you want to suggest a topic, then, I mean, if you want to suggest a topic to be pulled out of a hat to do a future Whose Gene Is It Anyway stream, send that to me on Twitter. And use the hashtag Whose Gene, and the ideas will be added to the hat if they're feasible. If you've got an idea for a topic for Gabe and mine, like for the Friday, for me and Gabe to discuss, you can send that to me on Twitter as well. If you haven't already, be sure to, like, subscribe, all that stuff. Be sure to check out my second channel, The Taste Emporium. There'll be a new video, hopefully by the end of the week. That's what I'll be doing. I'll be filming that tomorrow. So yeah, be sure to do all of that kind of stuff. I'll be sure to keep you guys appraised and updated on any updates in terms of the milk and egg plasmids as they get made. Any other updates, be sure to follow me on Instagram, Twitter, Facebook, all those things. I'm posting updates all the time. We've got some really exciting bio work. I've been doing some more work on the Spider Silk project. That'll be coming very soon. Very excited. Hopefully I'll be able to make some, I think next week, probably next Monday, rather than a Whose Gene, we're going to see if we can try and do some lab work, and like actually be in the lab and actually make some of the Spider Silk stuff. Not the silk itself, but like doing some of the gene work on the stream. That'll be a lot of fun. Be sure to tune in for that. And with that, I will leave you. I hope you guys have a good day. Stay safe, stay healthy, all that kind of stuff. You know the drill. Wear a mask. Don't lick doorknobs. Have a good day. Bye-bye.